Thank you very much for that introduction. Good morning, good afternoon, good evening, depending on what part of the world you're in today. My name is Bill Rushton. I'm a process chromatography support scientist with Bio-Rad. And today, Dan Yoshikawa and myself will be speaking about a mixed mode approach, an alternative to a three-step mAb purification process. I'm going to start by giving an overview of what we're going to discuss today. So initially, we evaluated 3 monoclonal antibody purification workflows of a mAb with a PI of approximately 8.6. The first is fairly traditional monoclonal antibody 3-step process. We had a protein A capture, followed by an intermediate polish using our Nuvia HRS resin, which is a cation exchange resin, and final polish using our Nuvia Q, which is a standard anion exchange resin. The second workflow was a capture using Protein A Resin, and then a polish using our Nuvia aPrime 4A, which is a hydrophobic anion exchange mixed mode resin. The final workflow was captured using Protein A and a polish using our CHT XT media, which is a calcium affinity cation exchange mixed mode media. Then we're going to discuss some of the alternatives to the traditional mAb 3-step process, focusing on the aim of time and cost reduction while maintaining comparable purity and yield. And then finally, there'll be a discussion on exploring functionalities of mixed mode resins, including buffer additives. What you see here now are the workflow overviews for the 3 workflows that were evaluated. For all 3, we had CHO cell culture supernatant that was captured over Protein A affinity resin. And then for workflow 1, as I mentioned previously, we had an intermediate polish using Nuvia HRS, followed by the final polish step of Nuvia Q, anion exchange resin. Second workflow, after the Protein A capture, we had a polish purification with the Nuvia aPrime 4A. And then the third workflow, after the Protein A capture, we had the polish purification using CHT XT. Now I'm going to dive down into the chromatography operating conditions for each process step. For all the workflows, a Protein A capture was done. And here you can see the binding buffer and the elution buffer that was used, as well as the flow rate was 300 centimeters an hour. The intermediate step for workflow 1 was a Nuvia HRS column. It was run in a 50 millimolar acetate pH 5, and then eluted using a 0 to 100% B gradient up to 1 molar salt. The Nuvia Q and the Nuvia aPrime 4As were both run in flow-through mode using a 50 millimolar tris, 50 millimolar salt pH 8 buffer. And finally, the CHT was done in a sodium phosphate pH 7 and a 0 to 100% B gradient up to 1 molar salt. And all these runs were performed using a Bio-Rad NGC chromatography system. Now I'm going to dive into more detail in each individual workflow. So again, here's the traditional 3-step workflow that we used for this process. So the Protein A capture followed by the Nuvia HRS intermediate purification, and the final polish with the Nuvia Q. Going to start with the Protein A capture. Here you see overlay chromatograms for the 10 runs that were performed using the cell culture supernatant. The overlays show really good consistency from each run. The 2 peaks that you see later, you have the intermediate wash peak, which is that smaller peak, and then the elution peak in the acetate pH 3.0 is outlined in the orange box. Now showing some of the analytical work that was done of the pool of the Protein A eluates. So all 10 runs were combined, and SDS page and HPLC analysis was performed on these. If you look at the gels, in lane 2 is the cell culture supernatant, and in lane 8 is the elution pool. You can see that there was a decent amount of cleanup. You don't have as much of the smear in the upper bands from the cell culture supernatant. And then for the HPLC analysis, we had a yield of 100% and our purity was 96.1, and total amount of antibody loaded was 125 milligrams. So now I'm going to continue on and talk about the intermediate polish and the final polish steps. In the upper left, you have the Nuvia HRS, which is a cation exchange. So the material was loaded in acetate pH 5.0, and then a 0 to 100% linear gradient to 1 molar salt was performed. The chromatogram shows in the orange box where the elution peak comes off during that gradient. With the Nuvia Q, the material was pH adjusted and was able to be loaded on directly to the Nuvia Q. The buffer was a 50 millimolar tris, 50 millimolar salt, pH 8, and this was run in a flow-through mode, which is indicated in the orange box. Now I'm going to discuss the final analytical work post-Nuvia Q. On the left, you see the SDS page analysis. In the left box is the non-reduced sample, and the right is the reduced. You can see there's not a lot of impurity bands present. SDS page analysis done on the right shows a purity of 100%, and the overall yield of the Nuvia Q was 97.1%, and the amount loaded onto the Nuvia Q was 35.5 mg. I'm going to begin talking about the two-step workflows that were performed next. Here you see workflow 2. Just as a refresher, it was a protein A capture followed by polish purification using Nuvia aPrime 4A, which is a hydrophobic anion exchange mixed mode resin. I'm going to talk a little bit about Nuvia aPrime 4A, a hydrophobic anion exchange mixed mode resin. The ligand itself has the quaternary amine, so it's going to have a net positive charge. So basic species, which are high PI target proteins or other host cell proteins, they will have a net positive charge as well. So you will see electrostatic repulsion. Acidic species, which will be low PI proteins, host cell proteins, endotoxins, viruses, etc., will have a negative charge, so they will have an electrostatic interaction. The chromatography can be performed in either flow-through or bind-Elute mode. You can also adjust your buffer pH and conductivity to impact the charge and hydrophobic interactions. And then you're able to fine-tune the selectivity and recovery of the target molecules with, as I mentioned, buffer pH and conductivity. Here you see the chromatogram of the Nuvia aPrime 4A run. The post-protein A eluate was adjusted to pH 8, and the conductivity was also adjusted to match that of the buffer used, which is 50 millimolar tris, 50 millimolar salt, pH 8. The orange box indicates the flow-through section of the column run, as this material was run in a flow-through mode. Here you see the SDS page and HPLC results from the Nuvia aPrime 4A run. On the left, the non-reduced and reduced SDS page show very high purity material. The HPLC results gave a purity of 99%. The overall yield off of the aPrime 4A column was 92.4%, and the amount of material loaded was 181.7 milligrams. Finally, I'm going to talk about workflow 3. This was a two-step purification workflow, starting with the capture of the Protein A resin, and then the polish purification was done using CHT XT, which is a calcium affinity cation exchange mixed mode resin. I'm going to talk about some of the properties of CHT XT, which is a calcium affinity cation exchange mixed mode media. The amino residues will interact with the phosphate sites in a classical cation exchange mode. They're able to dissociate with sodium chloride or with other buffering salts such as phosphate, or you can also weaken or dissociate with increasing pH. The carboxyl residues will interact with the calcium sites and form that calcium chelation. It is significantly stronger than the ionic interactions, between 15 and 60 times stronger, and you're not able to dissociate even with 1 molar salt. You're going to require phosphate to actually dissociate off of those sites. And then finally, you have the phosphoryl groups will interact also with the calcium chelation by the phosphoryl oxygens. Again, it is substantially stronger than the ion exchange. And you're again going to require phosphate to dissociate the phosphoryl groups. Here you see the chromatogram of the CHT XT run. So you had the material loaded in a 10 millimolar phosphate pH 7, and then it was eluted using a 0 to 100% B gradient up to 1 molar salt. And the orange box indicates the elution of the mAb off of the CHT XT. Here you see the final analytical results for them off the CHT XT column. On the left, you see the reduced and non-reduced SDS page analysis, both showing a high level of purity. And the HPLC results gave a purity of 99% and a yield of 100%. And the amount of material loaded was 125 milligrams. The final data I'm going to be speaking about is the host cell protein removal across this process. So the cell culture feed had 4,252 nanograms per mil of host cell protein. Host protein A were at 196.6. The Nuvia HRS brought it down to 23.7 nanograms per mil, and the Nuvia Q was at 3 nanograms per mil. When you compare that to the two-step processes, the Nuvia aPrime 4A resin was able to bring the HCP below the level of detection, and the CHT XT was able to bring it to 3.5 nanograms per mil. The results showing that the HCB removal was comparable across all three workflows. Now I'd like to discuss the mAb Purification workflow comparison summary. When you look at the three-step process, the Nuvia HRS had a step yield of 94.3%, the Nuvia Q had a step yield of 97.1%, for an overall process yield of 91.6%. When you compare this to the two-step processes, Nuvia aPrime 4A at 92.4% and CHT XT at 100%, the two-step processes give a slightly higher yield to the three-step process. When evaluating purity, the Nuvia Q had 100% purity, whereas the Nuvia aPrime 4A and CHT XT both came in at 99%, which would be considered comparable for these studies. I'm going to speak about some of the advantages to going to a two-step process versus a three-step process. You could end up with a higher yield versus a three-step process and comparable purity, as our studies have shown. It can allow you to have unique selectivity. You can also afford a single-step aggregate and HCP removal. It will reduce your QC testing, regulatory, and documentation burdens. You will need less suite time for your column operations, which will then lead to a cost reduction on operational units, labor, and capital equipment. I'm going to end my portion of the talk with this final summary slide. I hope I've shown that a two-step process using Nuvia aPrime 4A or CHT XT can purify with similar or better recovery and purity than a traditional three-step process. All these resins can be operated either in flow-through or Bind-Elute mode. All of them can provide effective separation of target from impurities, and you're able to improve process efficiency with fewer steps. I'd like to make a couple of acknowledgments within Bio-Rad. Jamie Greenwood, who performed all the experiments for this study, and Sharon Bola, who helped organize this entire project. And with that, I'm going to transfer this over to Dr. Dan Yoshikawa, who's going to continue to speak about our mixed mode resins.
Thank you, Bill. In this next segment, I am describing the mixed mode resins offering we have at Bio-Rad. This slide shows a list of our resin offerings, and you can see the categories across the top. And in the next few slides, we'll talk about the mixed mode resins Nuvia cPrime, Nuvia aPrime 4A, and the ceramic apatites. The figure on the left describes the PI-pH relationship and how the relationship affects the overall net charge of a protein. This can be used as a guideline for the selection of a traditional single-mode ion exchanger, whereby you may already have an idea of the PI, and therefore you can estimate the protein's overall net charge by adjusting the buffer pH. The figure on the right applies to the use of mixed-mode hydrophobic ion exchangers. Here we need to consider the hydrophobic interactions between the protein and resin. Increasing the conductivity of the elution buffer, while that minimizes the electrostatic interactions, this will also start enhancing the hydrophobic interactions. And protein molecules have their own inherent mixed mode nature as well to consider. This is all to say that we can leverage these multitude of interaction modes to achieve the selectivity, whether it be in binding in elution mode or even non-binding flow-through mode. Here is Nuvia cPrime, a hydrophobic cation exchange mixed mode resin. The hydrophobic moiety is the benzene ring, and this ligand has a carboxyl functional group. The charge state of the carboxylate can be changed by buffer pH. For example, if the buffer pH is lower than the pKa of the ligand, 4.8 in this case, the ligand is neutral, and so the resin would behave like a traditional hydrophobic interaction chromatography resin. Conversely, if the pH is on the basic side of the pKa, such as pH of 7, the carboxylate group would be fully ionized and negatively charged, thereby performing as a classical cation exchanger. Here is a case study demonstrating the unique selectivity of Nuvia cPrime. We're comparing Nuvia cPrime with the classical cation exchanger, Nuvia S, to separate two proteins of basic PIs, ribonuclease A, 8.7, and cytochrome C, 10.7. The protein mix was loaded onto the Nuvia S resin and a 30-column gradient of two buffers, buffer A and buffer B, as noted on the slide. And as you can see, the two protein peaks were not separated. Cytochrome C is retained slightly longer, and this is consistent with cytochrome C being more basic. When a protein mix was applied to Nuvia cPrime, that's the top figure, and using the same buffer gradient system, we now see two separated peaks. Ribonuclease A now elutes later, and this is due to a stronger hydrophobic interaction with the Nuvia cPrime resin. The next example is a workflow for the purification of adenovirus involving Nuvia cPrime, followed by Nuvia Q, a classical anion exchanger. You'll see nice sharp peaks off of both resins. If you look at the SCS page gel, harvest is in lane 2, and the large band is albumin. Nuvia cPrime takes care of the albumin, as well as a lot of the other proteins. Then Nuvia Q cleans everything up, and what you see in Nuvia Q Elute are 5 bands, which represent the 5 major proteins, which you find in adenovirus. This gave a final yield of better than 50%, which for a viral vector is good for the 2-step process. DNA was below level of detection, and full cell protein was about 2 nanogram per 10 to the 10 particles. Here is Nuvia aPrime 4A, hydrophobic anion exchanger. Bill had introduced this product in an earlier slide, and we'll go over it again briefly. As we saw with Nuvia cPrime, Nuvia aPrime 4A has the phenyl hydrophobic moiety. aPrime 4A has a positive charge on the ligand. Under neutral pH, we would expect high PI proteins to carry a net positive charge and therefore not bind to the resin due to the electrostatic repulsion. On the other hand, acidic species will be bound by Nuvia aPrime 4A, such as low PI proteins or nucleic acids, endotoxins, viruses. As mentioned previously for Nuvia cPrime, we would leverage the multitude of interaction modes to achieve our desired selectivity of the target. And the resin can be run in either Bind-Elute or flow-through mode. Here is a purification of a basic protein. Impurities are product aggregates, HCP and DNA. The PI of the protein is 8.45. Based on the principles we just discussed, at neutral pH, the molecule will carry a net positive charge, therefore electrostatic repulsion between molecule and ligand, and the molecule to be in the flow-through fraction with impurities retained on the Nuvia aPrime 4A column. The feed contained 100 millimoles of sodium chloride. DOE, or design of experiment, was first used to find the effective buffer pH that gives good monomer purity and recovery. This chromatogram shows the purification run using a packed column. We achieved 94% target monomer recovery with 99% purity. And noteworthy to point out is the shape of the flow-through fraction. You'll see that there's no tailing, and this can be attributed to the design of the particle's pore structure as well as minimal non-specific interaction between the molecule and resin. In this next example, we use an acidic protein, a monoclonal antibody with a PI of 6.9. This mAb preparation contained high levels of dimer and tetramers that resulted from the Protein A purification step. Nuvia aPrime 4A chromatography was used for the polishing step to remove the impurities. Again, DOE screening was first used. With DOE, we found the effective buffer pH and sodium chloride concentration that gave good binding of the mAb as well as elution. And what you see is the chromatogram of the purification run using a packed column. First, using a column enabled us to determine the dynamic binding capacity. As for the purification, the bound mAb monomer was eluted at pH 6 with high purity. The impurities that remained on the column were then stripped with low pH. I'd like to make a brief comment regarding the buffer additives that can be used in mixed mode chromatography. Additives can be used to modulate the various modes of interaction between the target molecule and the resin. And these modulations can be leveraged to achieve optimal target purity and recovery. This is a list of common buffer additives often used in protein chromatography. Arginine is gentle and often used for minimizing non-specific interactions. Propylene glycol is found to enhance electrostatic interactions. Glycine has the opposite effect, enhancing hydrophobic interactions. Guanidine hydrochloride and urea have strong effects on protein and are often used for stripping strongly bound proteins during column regeneration. Our third mixed mode support, ceramic hydroxyapatite, or CHT. Bill had introduced CHT in the earlier section, and we'll go over it again here briefly. What's shown here is CHT's interactions with a variety of biomolecules in cartoon form. On the left, you see the surface of CHT with calcium ions and phosphate groups. And what's shown is the interaction of a protein with positively charged amino groups on its surface and interacting with via classical cation exchange with the phosphate groups on the surface of CHT. Like classical cation exchange, this interaction can be dissociated with either sodium chloride or with buffering salts like phosphate. You can also weaken or dissociate this interaction with increase in pH. The middle is showing the interaction of negatively charged proteins with the calcium ions on the surface of CHT. These are metal affinity interactions with clusters of carboxyl groups interacting with the calcium ions. This interaction is very strong. Similar to IMAC, dissociation requires a competitive inhibitor, and for CT, we use high levels of phosphate. On the right is interaction of highly negatively charged species, such as nucleic acids and negatively charged enveloped viruses. This interaction is also very strong and requires high levels of phosphate to elute the species off the column. CHT is considered by many to be the universal polishing resin, and what you see in this table are the common impurities that you find in a process feed stream. On the right are the observed clearances that one sees typically with CHT. Here's a case study involving monoclonal antibody purification with CHT XT, removing aggregates. The mAb has a PI of about 9.3. On the left is an HPLC SEC chromatogram of a load going across the column, and you see the load contains about 28% aggregate. The middle shows the purification of the mAb across the CHT column using a sodium chloride gradient. The large single peak is the monomer, and aggregate appears in the strip. The right shows the SEC of the elute coming off of the CHT column. The aggregate levels have been reduced from 28% to well below 1% in the final elute. In this particular run, a high level of purity was achieved with about 70% yield. Here's an example of virus purification, Polio virus, using ceramic apatites. Here are two chromatograms, one from CHT type 2 on the left and CFT on the right. CFT is ceramic fluoroapatite. This is material where the hydroxyl groups in CHT have been replaced by fluorine atoms. What you see in both chromatograms is very good separation between infectious activity in red and the bulk of the absorbing material, 260 to 280 on either product. The yields were very high, 88% with CHT and about 100% with CST. The final purities were also high, better than 90% in both cases, which is really good for single-step purification. In summary, method development for mixed mode chromatography is intuitive. Essentially, you're already using the techniques with the single mode traditional resins. The same concepts apply in mixed mode chromatography. You're just implementing them in a single resin. These are examples we discussed: ion exchange and hydrophobic interaction pairing for the Nuvia cPrime and Nuvia aPrime 4A, and ion exchange and metal affinity pairing for the ceramic hydroxyapatite. We mentioned that DOE is often used for initial condition screening to determine, for example, the pH and conductivity to achieve good purity and recovery of the target. We gave examples of additives that can be used to modulate interactions. The columns can be run in either Bind-Elute or flow-through modes, depending on the purification requirements. And Bill presented an example where mixed mode can streamline a process by eliminating steps. Please visit us online where you can find further details on all of these topics that were discussed. Bio-Rad offers a variety of process chromatography resources. We have a global network of experts ready to support you with your method development. We can assist you in optimizing steps and supporting you all the way from the lab up through scale up, and this includes process transfer and regulatory procedures. Check out our online resources at this link, where you can find a variety of product information, including useful packing tools. You can request resin samples, and you can contact your process specialist for further help or information. One final point: the image you see are our screening tools. They are called the Foresight pre-packed tools and are a line of products that include 1 mil and 5 mil pre-packed columns, 200 microliter, 600 microliter robo columns, and plates. Thank you for joining us today. We will now move on to the Q&A portion of this webinar. A few questions are coming in. Here is the first question. What two-step workflow would you recommend for an acidic mAb?
That's a good question, Dan. I think I'll take this for you. We found that a combination of Nuvia cPrime, Nuvia aPrime, and CHT are all able to bind acidic mAbs with great salt tolerance and with a decent binding capacity up to as high as 150 millimolar salt. If you'd like, we have additional application notes that we can email you for further details, or you can visit bio-rad.com/library for these as well.
Okay, thank you, Bill. Here's another question. Is CHT preferred over other purification methods for virus purification? Let me take that one, Bill. We have several case studies using CHT and single-step purification of viruses. We can also provide case studies using Nuvia cPrime and Nuvia Q. It's a two-step purification of Adenovirus. And we have 2 Ion Exchange Resins, Nuvia Q, used for single-step purification of retrovirus, and Nuvia HPQ, used in single-step purification of H1N1. And those are to name a few. Furthermore, these application notes are available on our website, Bio-Rad.com. Another question has come in. Where can I learn more about how to start DOE studies with additives? And that's, thank you for that question. I can take that. We can provide application notes as a guideline on where to start. You can obtain those information by emailing us at process@bio-rad.com. That's process@bio-rad.com, and we can provide you all the details. like we have one last question that's coming to the queue. The question is, do you have viral clearance data for Nuvia aPrime?
Dan, since I've had a lot of viral clearance experience in my day, I would be happy to answer this question. Yes, we have a technical note and a webinar on Nuvia aPrime that details our viral clearance that was done. It was initially was done in a DOE approach where we use spin columns to evaluate the ability of aPrime to clear minute virus of mice. From that point, we then scaled up to our 5 mil foresight columns and reevaluated and confirmed these conditions at a larger scale. We also performed some studies with CHT XT at the same time, and both gave very good clearance of viruses.
Thank you, Bill, for the nice detailed response on that. Okay, everybody, thanks again for joining us today, and please be sure to email us at, again, the email address is process@bio-rad.com, and with that we can provide any information you need, answer all questions you might have. Thank you, Bill. Thank you all.
Thanks, Dan. Have a good day.
Good night.